Hi, everyone. My name is Valérie, and I would like to welcome you to our virtual workshop on peptide modeling and docking. Please note that you're muted upon entry. Questions can be asked via the Q&A section. However, please note that we will be answering all the questions after the workshop via email. We'll also be sending out the video recording of the workshop via email to everyone who participated today. So that being said, let's get started. So here's the outline for our peptide modeling course. So typically this course has 5 sections, as you can see on my slide here, but I've selected 2 topics for today since we only have 1 hour together. So in the first section, we'll be covering protein-peptide interaction analysis. So we'll be loading in a complex of MDMX with a P53 peptide, and then we'll be looking at how do we prepare the structure and do some basic analysis. So looking at the interactions between the two, the surfaces, also calculating some properties of our peptide. In the second section, we'll be looking at docking. And actually there's 2 subsections here. We'll be looking at how we can use both the protein-protein docking application in Mo, as well as protein-ligand docking to dock our peptides into our receptor. So the sections that are grayed out, we'll be skipping today, but of course you have access to this PDF. So if ever you want to click through these examples, of course you can go ahead and do so and email us if you have any questions. So let's get started. So the first topic, as I said, is going to be the interaction analysis of a protein peptide complex. So what we'll do here is we'll start by loading and preparing our structure, and then we'll look at different tools that we have in Mo to analyze and identify the contacts that lead to the binding of our peptide. And then we'll also look at some protein surfaces, some patches to really understand how these things are actually interacting. Finally, we'll look at some peptide property calculations. So that being said, let me minimize these slides here and we'll jump right into Mo. Okay, so the first thing I'll do is to open the structure of our complex. So this is going to be MDMX with a bound peptide. This is a fragment of the P53. So for that, I'll go to my right-hand side button bar. Of course, that's this set of tools right here. And I'll click on Open to access the Open panel. I have it right here. This is going to be 3DAC.pdb. So it's a PDB structure. I'll go ahead and click OK to open the structure. And of course, I'm prompted with this Load PDB file panel since Mo recognizes this as being a PDB file. Often I use just the defaults here, but I happen to know that there's actually 2 copies of this structure in the PDB file. So there's 2 copies of MDMX in complex with P53 peptide. So what I'll do is in the symmetry pull down here, I'll just go ahead and select biomolecule assembly. That way we'll only select or only load in 1 copy of the complex. And I'll just go ahead and click OK with all the defaults. Now, as I do that, you'll see the structure appear on the screen. And of course, it's color coded by secondary structure. So we can clearly see all the alpha helices here in red and the beta strands in yellow. And as I rotate around, we can already guess that this must be the peptide that's bound here. And this must be the protein because it's clearly larger. Now, the very first thing I always do when I load in a structure is to prepare it. What do I mean by that? Well, it's a crystal structure, so there's likely no hydrogens, no charges, and there could be some other issues. Sometimes there's some missing side chains, some naming issues with some of the atoms and things like that. So what I'll do is I'll go back to my right-hand side button bar, and just below the open button, we have the quick prep button, which opens our application for preparing structures. So the quick prep panel is separated in 3 sections. You'll notice the top section is for cleanup and preparation of the structure. The middle section is for setting tethers and constraints. And the bottom section is for refinement. Now, in this case, I'll use most of the defaults. The only thing I'll change here is this option here where it says fix. So by default, it actually fixes all the atoms that are far away, so more than 8 angstroms away from the ligands. But that means small molecule ligands, which I don't have here, right? I just have a peptide, that's my ligand. And so by default, it would actually fix everything, which clearly I don't want to do. So I'll toggle that off and I'll just go ahead and click OK. Now, while this is running, first of all, you'll see the progress report in the top left corner, but I'll just reopen quickly the quick prep panel to tell you a little bit more about what's going on. So first of all, the preparation or cleanup section uses 2 different tools in Mo. The first one is structure preparation, which will take care of a lot of the issues I mentioned previously. So adding in hydrogens, charges, missing side chains, if there are any missing side chains. It also builds in loops if they're short enough. if they're missing loops. So in this case, there's no missing loops, so that won't be done. And so basically, that's what structure preparation does. Then we use something called Protein 3D, which is another tool in Mo, which will optimize the ionization state of our bound molecules, if we had any, as well as the side chains of our protein. It also optimizes the placement of the polar hydrogens to optimize the hydrogen bonding network. Okay, so moving down, here we set some tethers by default on the receptor. That's just so if we do some energy minimizations, which we will do, we won't deviate too much from the initial crystal structure. By default, like I said, we fix atoms far away from the ligand. That's just done for efficiency, really. And so here we skip that step. And finally, we do an energy minimization as the final refinement step. Now, that being said, I'll cancel this. I already ran it, and I can see in the top left corner, there's no text left, which means my structure is now prepared. Okay, so as I mentioned, we can already guess that this is the peptide, but of course, a better way to get a full view of what we have in our system is to use the system manager, which we can open by clicking on the blue system button at the very top of the window. I'll just go ahead and attach that. It's more convenient for me to actually always see what I have on my on the side of my Mo window. Now, in this case, I only have one tag. That would be the 3-DAC structure. But of course, the system manager allows me to manage multiple structures. If I wanted to load multiple in here, they would all be listed right here. Now, right now, I see I have a receptor chain and a solvent chain. Turns out I'm expecting 2 chains here, right? Because I have the peptide and the protein. But that's just because I'm using the default settings in Mo, which is the basic view. I'll just change that to the extended view. That way, I can really see that I have 2 chains, the receptor chain and the protein and the peptide chain, as we'll see. Okay, so just a quick review of the system manager going from left to right. The open circle is for selecting. So if I click on one of these, I can select the atoms from that set, from that chain. So I selected in here the second one, and so that's my peptide. The green A means active. That means right now, if I hit minimize, everything in the system will be minimized, including these solvent chains. So these water molecules, which clearly I don't see currently in my Mo window, but they're still active. So they would be used as well. When I click on it, it becomes an I, which means inert, or inactive, which means they would be ignored. Moving along, we can hide and show different chains by clicking on them. And then we have different tools to change the rendering. So the color, the bond, and atom display, as well as the ribbons. And finally, this S button, I'll come back to that a little bit later on. That allows me to create or modify existing surfaces. The red X, of course, is for deleting chains, which I won't do today. Okay, so to have a clearer view of my system, it might be a good idea to color both chains differently. That way I really can know which one is my peptide, which one is my protein. Like I said, these colored boxes allow me to do that. But just a quick trick, if you click on the first one on the tag here, you can actually automatically assign a different chain color to each chain. That way you don't have to manually go select a different color for each. Here there was only 2, but if you had this really big complex, it would be useful to do that automatically. So now I can clearly see that my receptor, so MDMX, is this orange chain right here, and this one here, that would be my peptide. So let me just go ahead and rename these so we can keep track of what we're working with. So this is MDMX. So I'll call it MDMX. And if I make this a bit wider here, you'll notice that it's been renamed. There we go. And I'll rename this one P53. There we go. So now I clearly see what I'm working with. Now you'll notice that both of them are annotated as being receptors, right? Because they're made out of amino acids and there's a cutoff. If it's more than 10 residues long, it's annotated as a receptor. If it's shorter, then it's annotated as a ligand. However, I can customize that. So this peptide here, I want to consider it as a ligand. So I'll go ahead and select it. And then if I go to the right-hand side button bar, click on ligand, I can do choose ligand. And then you'll notice in the system manager, it gets reassigned as a ligand. Now, the reason I want to do that is it will give me access to a few more tools, right? So a peptide is somewhere between a protein and a small molecule ligand. So it's made out of these amino acid building blocks, of course, but it's a shorter fragment. So we can also use some of the ligand tools. And so by annotating this as ligand with the choose ligand tool, it will allow me to have access to a few more tools from the small molecule toolbox. Okay, so now that we have this, we understand what we have in our system, let's start looking at the interactions between the peptide and the protein. I'll show you 3 different ways we can look at the interactions. So the first one, we'll go ahead and hit site view to zoom into the active site. So zoom into the peptide And basically site view shows me everything within 4.5 angstrom radius of my ligand, right? So my ligand being the p53 fragment. I'll just go ahead and display the ribbon again. So that's my peptide. And now we can see all the water molecules as well as the side chains of MDMX within that 4.5 angstrom radius. So there's quite a bit of water molecules here. So it might be a good idea to have a quick look at them. And as I zoom in, we can see that a lot of them are interacting either with the protein or with the peptide. But if I took some time to actually look at each of them, I would notice that none of them are actually directly bridging any interactions between the peptide and the protein. In this specific context, of course, in another structure, perhaps some of the molecules are actually quite important, in which case we would want to consider these. But in this case, because none of them are actually bridging interactions between my peptide and my protein, I'm actually going to choose to hide and make those inert. So we won't be using those throughout the course today. So to do that, I'll go back to my system manager and I'll click on the solvent chains here. They become hidden, but also inert. Okay, so let's go back to the MAMO window and have a look at these interactions. So you'll notice that there's these dashed lines here in different places, and those are telling me where there's hydrogen bonds between the peptide and the protein, but also we can see some within the peptide itself, right? This is an alpha helix here, so clearly there's a lot of hydrogen bonds within the peptide itself. But what I'm interested in right now is to look at the interactions between the peptide and my MDMX structure. So on this side here, I see a pair of interactions with this residue right here. If I select it, I can see that's glutamine 68. If I move to the side here, I can see some more interactions. So one right here between this tryptophan and this residue right here. If I click on it, I see that's methionine 50. And further on this side, there's an interaction with this tyrosine right here, and another pair of interaction with this, this lysine right here. Now you saw that as I clicked on different residues, I got the information on the footer, which residues were involved. But of course, I could also choose to just add some labels from the footer to all the residues that are currently displayed. That way I'll always be able to see which residues are involved. So these dashed lines are showing me where there's hydrogen bonds, and you'll notice that there's also non-standard interactions which are displayed in that way. So for example, here I see a dashed line with this interaction with the methionine as well. One last note about these dashed lines, you'll notice there's a cylinder in the middle. If I zoom in, you'll notice that this cylinder is bigger than this one. And that's simply telling me what's the relative strength of these hydrogen bonds. So basically this glutamine is making a stronger interaction with this backbone nitrogen than with the side chain of this threonine right here. Okay, so that was the first way we can look at the interactions between the peptide and the protein. The second way is using the ligand interactions diagram. So let me just zoom out a bit so we have a clearer view. And I'll go to the right-hand side button bar and click on the ligand menu to open up the ligand interactions panel. So the ligand interactions panel gives me a schematic view of exactly the same thing I have in my Mo window. So it tries as best as it can to position the ligand, so in this case, the peptide, in more or less the same orientation as we have it in the main Mo window. Of course, this is a pretty big ligand that we have here, this peptide. And so, you know, it's a little bit hard to display that in 2D, but basically you can tell right away that, you know, the phenylalanine right here is this one, the tryptophan is this one, and so on and so forth. So it allows you to really compare the 2D and the 3D view. So what does this tell me? Let me make it a little bit bigger. So as I mentioned, we have a 2D view of the, or 2D schematic of the peptide. Around that, we have this dashed line, which is showing us the relative position of the surface. And we have these circles here, green and pink, which are showing us the residues that are involved in the binding pocket. So those are the residues on our protein receptor, so MDMX in this case. Now you'll notice they're color coded, so the green ones are the greasy residues and the pink residues are polar. If they're charged residues, they have a blue circle around them when they're positively charged and a red circle if they're negatively charged. And in this case, my binding site doesn't contain any negatively charged residues. Now, in addition to that, of course, we can pick out the hydrogen bonding that's going on by looking at these arrows right here. So these dashed lines with the arrows tell me where there are hydrogen bonds. And so we're getting the same information we saw in 3D, but now displayed in 2D. So we have the glutamine here, the tryptophan interacting here, this interacting, and this pair of interactions as well. In addition, you'll notice there's these blue halos on some of the atoms of my peptide. This is telling me where there's solvent exposure, right? So right away, I can tell that some of these residues, for example, the phenylalanine, the tryptophan, and to some extent, these two leucines here and here don't have so much blue on them, not as much as the other side chains anyways. So these appear to be some residues that are on the surface, right, on the contact surface between my peptide and my protein because they're not solvent exposed. Whereas these ones here, quite a few of them actually, appear to be quite solvent exposed. So if I zoom out and I rotate here, that's pretty clear. All this part of the peptide is completely solvent exposed. So the key residues that are making contacts, we can pick them out both in 2D and 3D, are the phenylalanine, the tryptophan right here, this leucine, and to a lesser extent, but still this leucine right here. All right, so that was the second way we can look at the interactions between our peptide and our protein. So let me close this here. The third way is by using the protein contacts application. So I mentioned the peptide, we can use both small molecule tools and protein tools to analyze them. And so in this case, what I'll do is I'll go to the protein menu at the top here, and I'll go and select our protein contacts application. So the protein contacts application, I'll just move it to the side here, contains basically 3 sections. So at the top, we're going to set up what are the 2 sets of atoms we want to use to compute the contacts or the interactions. In the middle section, we'll get a list of all the contacts, and at the bottom, we can filter those out because, of course, in protein-protein interactions, there can be a very long list of these contacts. So let's set this up. So for set A, I'll set this to be my protein. So I'll select MDMX. For set B, I'll go ahead and select my P53 peptide. And just like that, I'll go ahead and click search to compute the list of interactions. Now you'll notice there's different types of interactions we can compute, and here's the list of the ones that were computed. We can compute distance interactions. Currently, they're not displayed. I'll come back to that in a second. Covalent interactions, arene interactions, ionic interactions, metal interactions, as well as hydrogen bonding interactions. Now in the list here where it says type, this is telling me the type of interactions that were found. So H here stands for hydrogen bond, and the I at the top stands for ionic. If I select one of these in the list, for example, the first one, which appears to be the highest energy, and I could sort them by energy as well, and I look in the Mo window, I see that it's been highlighted with these halos here, just to really easily point out where this interaction is. So here it's a peptide. It's pretty easy to point out the interactions, but this can be used on a much larger peptide or even on protein-protein complex, and quickly allows you to find where the key interactions you're interested in. So here I selected one, but of course I could go ahead and select all of them in the list. And just like that, they've all been highlighted in the Mo window. Now, coming back to the types of interactions, I'll clear this. I mentioned there's also distance interactions. By default, we don't toggle those on just because when you're calculating the contacts between 2 big proteins, the list can quickly be really, really long. But of course, here I do want to know about these distance contacts, or basically van der Waals contacts. So I'll toggle that on, and automatically the list becomes much longer, and all these D interactions, those are distance or van der Waals contacts. So I can see there's quite a few residues that are interacting via distance contacts. Now, you'll notice there are 2 tabs here, so I can look at the contact list, but I can also view the same information as a plot view. Now, on the plot view, on the y-axis, you have the residues from set A. So in this case, from MDMX, my protein. And we can see there's about 100 or so residues in that protein, so a fairly small protein. On the y-axis, I have the residues from my set B, so from my peptide in this example, so about 12 or 13 residues in this case. Now, the way to look at this plot here is to look for lines of dots. So anywhere you see a vertical or horizontal line, for example, I'll select this set of dots right here. That means I have a hotspot. That means I have one residue, in this case on my peptide, which is interacting with multiple residues on the binding partner, in this case, MDMX receptor. So if I select these, of course, they get selected here. If I go back to the contact list, they've been now selected here, and I'll just go ahead and resort this by the position in my peptide. And I can see that this hotspot happens to be the tryptophan 23 residue. And of course, because I've selected the interactions now, they've been highlighted in the main Mo window. So easily, I've just pointed out that this tryptophan residue from my peptide is making a number of different contact interactions, as well, of course, as this hydrogen bond interactions that we had noted before with the backbone of methionine 50. So certainly a key contact here, or key residue, should I say, in this peptide. So if I were thinking about making modifications to this peptide, that's probably something I don't want to play with because it's making this really nice hydrogen bond and a whole bunch of different contacts as well with the protein environment. Now, if I go back to my panel, I can see that tryptophan 23 is not the only residue that's making a number of close contacts. I can scroll and I can see there's a few others: phenylalanine 19, leucine 22, as well as leucine 26. And those are exactly the same residues I had identified both in the 2D view with the ligand interaction diagram, as well as in the main Mo window. So basically, those are the three ways you can look at for the interactions between your peptide and your protein, given a complex that you have, so a crystal structure or even a dot structure of your peptide. So that being said, let me just close this here, and we'll move on to the next thing I want to show you, which is how to compute properties for your peptide. So again, because we can use both the small molecule tools and the protein tools, there's two ways we can compute some properties for a peptide. The first, we'll look at the ligand properties. So from the right-hand side button bar, if I click on ligand, I can choose ligand properties in the middle of the menu. As I do that, this is an on-off switch. As I toggle this on, you'll notice in the top left corner, there's some properties that were listed. So things like the weight of the peptide, TPSA, log P, and so on and so forth. So these are 2D properties, of course, but if I hit minimize on the right-hand side button bar, so it will minimize my system, I'll get additional properties listed: the strain energy, the solvation, and the affinity. So if I were interested in making modifications to this peptide, I could keep track of these properties. Now, as I mentioned, we won't be covering that today, how to make modifications to a peptide and how to optimize the sequence of a peptide. However, it is in the course material. So I'll just take a second to point out, if you go to the help menu right here, that you can actually go to our courses page where you can go and select any of the courses you want, including this one, to download the PDF. So that's the first way we can calculate some properties. Now I'll go ahead and turn these off, and I'll go ahead and calculate properties using our protein properties application. So under protein, we can select properties, and this opens up our protein properties application. It's separated in two sections. So the top here allows me to configure which set of residues or which protein is going to be used for the calculation. So here, this is the receptor, but of course I could do the same calculation on my peptide by selecting ligand from the list, since my peptide is a ligand. And then I can configure the different settings or conditions at which I want to calculate the properties. Below this, we have a list of 109 different properties to choose from. And as you can notice, some of them are pre-selected, the ones in blue, 28 of them actually are pre-selected. So that being said, I'll go ahead and click calculate to launch the calculation. It will calculate all 28 of these properties for me. As I do that, it automatically switches from the property calculation tab to the property viewer tab, where I can view the results. So these are all the properties that were calculated. Now you'll notice that some of them have an empty field here, but a plus sign here. So if I click on the plus, I get a list. These are per residue properties. And so I get a list of the property values. For example, in this case, the hydrophobic residue patch area for each of these residues. When I'm looking at per residue properties, though, I tend to go to this option right here, residue table, which allows me to get a full list of the residues. In this case, the list is short because it's the peptide. And for each of the peptide residues, I get the values of the properties that were calculated. One last note about this before I move on. There's also a sample mode here, which can be useful. So the sample mode will allow us to calculate these properties, not just on the single static conformation of our protein or our peptide, but actually do some conformational sampling and a sampling of the pronation states at the same time to get a better representation of our system. So that being said, the last thing I want to show you before we move on to docking is how to generate surfaces. So we looked at the interactions, right, of the peptide with the receptor environment. But another thing that's really useful is to see, what's the context of this binding? Is it binding in a deep pocket? Is it a shallow groove? What does the contact surface between the two actually look like? So to do that, what we'll do is we'll go to our right-hand side bun bar. And I'll click on the surface menu right here. And from here, we have a list of different protein surfaces to choose from. So I'll start with the protein surface, which will draw a Conley surface around the entire receptor surface or protein surface. So in this case, around my MDMX structure. So by default, it's colored white. And if I rotate around, I get an idea of the shape of this protein. But more importantly, of how this peptide is actually sitting on that surface. Right. So if I rotate it, I can see that it's actually a fairly shallow groove here. It's not a deep pocket as you would think of for small molecules. Like a lot of peptides, it sits on sort of a groove on the surface of my protein. Now, currently it's colored white, but I can also map on some colors here on some properties or the atom type onto the surface. And so that's what I'm going to do right now. So if you remember at the very beginning, when I talked about the system manager, I mentioned the S button allows us to create or configure or customize the rendering, let's say, of the surfaces. So if I click on it again, I can see that now I can either create some new surfaces or modify my existing protein surface. That's what I'm going to do right now. So where it says color, instead of constant white color, I can choose from any of the options here. For example, coloring by electrostatics, by B factor. In this case, I'm going to choose lipophilicity. So if I click on lipophilicity, you'll notice that it gets colored in two colors, either pink or green, or somewhat white if it's somewhere in the middle. So what does that mean? Well, it's the same thing as we had for the coloring scheme in our ligand interactions diagram. So green means greasy or hydrophobic, and pink means polar. So if I'm looking at the binding groove for this peptide, I can see that it's actually pretty green here, so mostly hydrophobic. If I zoom out, I can see there's regions, for example, here that are more polar. Now, the coloring is really useful. However, it doesn't really allow me to know if there's regions that are specifically or particularly polar or charged or hydrophobic. And so we have a tool to not just color the entire surface, but really point out what are the key regions on our surface that are very charged or very hydrophobic. And so what I'll do here is I'll go back to my surface menu and I'll select the protein patches option. The protein patches will generate three types of patches: green patches, which are regions of excess hydrophobicity; blue patches, for example, here, which are regions of excess positive charge; and red patches like this one, which are regions of excess negative charge. So here they were simply drawn or mapped onto the existing protein surface. And so I can see that some regions, for example, this one that was pink, yeah, it was pink, but it's not significant, right? It's not very charged. And so it's not colored with these patches. However, what this allows me to point out here is that this binding groove where we have the peptide, that's significantly hydrophobic. So it makes sense. We know that often proteins come together, bind together, or proteins and peptides bind together by covering large hydrophobic patches. And that appears to be the case here. Again, thinking back on what we learned with the ligand interactions diagram, makes sense. We saw that, it was a tryptophan, a phenylalanine, and a pair of leucines that were in close contact. We saw that with the protein contacts application as well. And so it makes sense. These are all hydrophobic residues. And so they're leaning against this hydrophobic patch on our MDMX surface. Now, of course, if I rotate, I can have a look at what else is on this protein. And so on the back side, there's a fewer of these patches, some negative patches here, a bit of a hydrophobic patch here. But the most significant hydrophobic patch is clearly this one, where our peptide actually binds. Okay, so before we move on to the next section, so docking, I just want to show you one last type of surface that you might find useful. So let me just clear these. And the next surface I want to show you is something called interaction surface. The reason I find interaction surfaces really cool is that they really allow you to see the steric fit, right? So in the last example, so the last type of surface, we could see how that peptide was fitting on top here. But it was hard to see where there might be some opportunities to make some modifications to that peptide if that was our objective. So with the interaction surface, if I zoom in here, and maybe I'll make this a bit transparent, what we see is that it's actually a really, really tight fit. So let me just display maybe the hydrogens on my peptide. And so with the interaction surface, basically what we're looking for is for the atoms to be touching the surface. So if I rotate, let's look at this phenylalanine. It's hugging that surface right here. Same thing with the tryptophan here. It's following the shape of this pocket really nicely. And so these are regions where there's a really tight fit between the peptide and the protein environment. And so if I were thinking about making modifications, what I would be looking for to improve, if I wanted to improve the affinity, basically, would be regions where there's excess space. For example, here, perhaps I could add something in this position. So when the peptide or the ligand isn't touching this interaction surface, this mesh, those are regions where there's maybe some room to grow to optimize the fit of our ligand or of a peptide for the protein environment. Again, we won't be covering making the modifications in the pocket today, but that is in the course material. So if you're interested, go download our course, and you can always email us if you have any questions. Now, that being said, let me just maybe save this so we have a backup copy. So I'm going to say, I'll clear the surface first of all, and I'll hit site view once more, and I'm just going to save this file. That way I can come back to it, and we'll move on to the next section, which is docking. Okay, so I'll bring back my slides. So we finished this section right here. So protein-peptide interaction analysis. And now we'll be looking at protein-protein docking. So as I mentioned, we'll be covering two different methodologies for docking peptides in Mo. Today, we'll be looking at using the protein-protein docking application, but also looking at using the protein-ligand docking. In other words, we can consider our peptide both as a protein or as a small molecule ligand, but there's some things we have to consider if we're considering it a small molecule ligand, and I'll come back to that in about 10 minutes. Okay, so let's start with protein-protein docking. So the idea in Mo for protein-protein docking is that we initially use a coarse-grained representation of the proteins. Because you can imagine two proteins, in this case, it's going to be a peptide, but imagine in general two proteins, both can have thousands of atoms to them. And so the search space of how these two big molecules can come together is actually quite large. And so it's actually quite time consuming to consider all the possibilities of how these two entities can dock together or can bind together. So by using a coarse-grained representation, the idea is instead of considering initially thousands of atoms, we use a bead model where we represent each residue with 1, 2, or 3 beads, depending on the size of the residue. And these beads are of different charges and sizes. And so instead of having thousands of atoms, we can get this coarse-grained representation, which is made out basically of hundreds of points instead of thousands. Some advantages with that is that we get then a smoother potential energy surface, and also it's possible to do much faster energy calculations and minimizations. And so the overall idea, without going into all the details, is that we use these bead models, so this coarse-grained approach, use a fast Fourier transform approach to generate tens of thousands of initial poses. Once we have that, then we increase the level of complexity of our model on fewer poses at every step until we get a final 1,000 poses that we refine with a full GBVI solvation model. And so in the end, we output just the best poses, of course, to the output database, and then we can look at those poses. So that's the basic idea. I'll come back to this while we're running through the example, but we can also use restraints to restrict the binding site. So in this example here, this is an antibody, and we're restricting the binding to the CDRs where we would know where we know the antigen should be binding. But similarly with our peptide or any protein-protein interactions, often we have some prior knowledge about where roughly things are binding. And if we have that, we should always use that to our advantage because it reduces the search space. So let's have a look at that in Mo. So what I'll do, I'll just redisplay my ribbon here, and I'll go ahead and open the docking application. So I'll go to the main protein menu, and I'll select protein-protein dock from the bottom of the menu. This opens up our unified docking application. Right now, because I use the protein doc menu, it opened in this protein tab right here. But this same panel allows me to do general protein ligand docking, template-based docking, docking to electron density, reaction-based covalent docking, as well as protein docking, and external is for using external programs. So let's look at the protein docking to start. So I'll just move this to the side and we'll set it up together. So in the top section, this is where I'm going to define what's my receptor. So in this case, of course, MDMX is my receptor. So that's good. I'm going to be using what I have in my MO system. Here, receptor atom, that's okay. That's what I want to use. However, where it says site here, this is where I can restrain where I'm going to dock. So by default, I'd be docking everywhere around MDMX. And let me just show the ribbons on the full structure again. So I would be generating potentially some bound poses where this peptide is actually bound on this side or this side. If I had no a priori knowledge, that might be the thing to do. But here, I'm going to assume that I know it should be binding in this region here. So I'm going to set the site to be just the pocket atoms. In any of these panels, if you see the question mark, you can always hit the question mark button. It's just going to highlight briefly in the MO window which atoms are involved, just as a quick check that you've actually selected what you wanted. So that's what I'm going to select. Of course, in a case where I'm starting with an apo structure of my protein, I wouldn't have pocket atoms, but I could go manually select residues that I know are important, a region of my protein I know is important for binding, and then I could select something like selected atoms from the pull down. But in this case, we'll dock on the pocket atoms. That makes sense. Then here we can configure what's the ligand. So in this case, of course, the ligand atom is the peptide. So that's okay. We can set that to be the defaults right here. Now at the bottom, and the ligand site, I should mention the ligand site. So just like for the receptor, we can define a region of our receptor for docking. We could do the same thing on the ligand, right? So if I want to focus my bound poses only on this portion or this side of my alpha helix, I could do that. In this case, I'll just leave it as is. So maybe we'll have different orientation of that helix bound to our protein receptor. At the bottom, we have some extra options to restrict even further the bound poses that we're going to get. So in addition to restricting the sites, if we have an antibody, we have this option to restrict the sites to the CDRs. Of course, this is not an antibody, but we do have this option, which I will toggle on, which is to use hydrophobic patch potential. If you remember in our analysis, we saw that this peptide, the binding pocket where we want to dock, is actually very hydrophobic. So basically what this will do is add an additional term, which will favor poses where we're bearing hydrophobic patches, such as the one that we have in our complex here. So that makes sense to use, and we'll run it with the default. Maybe I can just reduce the poses. Typically, I leave this to the default. Although here this is a peptide, I might reduce it a little bit, but certainly not as much as this here. I'm exaggerating a little bit for the purpose of our demo today. And then basically, once you're ready, you can hit run and it launches a calculation. As you do that, a database viewer opens up. This is where we're going to have the results. And in the top left corner, of course, we have some progress report. I'm actually going to cancel this calculation in a second because I do want to get to the next section and we only have maybe 20 minutes left. But basically that's it. I would let it run and get the results in this database. Before I show you the results though, I just want to show you, there's also this batch button, which is very useful. So here I'm docking one peptide, that's okay. I could run it on my laptop and let it run, no problem. But let's say you had multiple peptides you wanted to dock, maybe even a database of peptides. What you might want to do instead of running it locally on your laptop is use this batch button to generate a batch file, which you can then launch from command line. If you have the HPC framework set up, you can even launch it directly onto your queuing system. So that being said, let me just stop this calculation. Like I said, I'm actually going to show you some previously run results here in the interest of time. So I'll go to the open button and I have some pre-calculated results. So the output of this run would look something like this right here, where, of course, the database viewer contains the results. It looks a little bit like an Excel spreadsheet, but you can see that already that it contains the actual structures, right? So it's much more than that. It contains structures of our molecules, in this case, the bound pose of the peptide together with the receptor. In addition, this S field here, that contains our final docking score. Docking scores, energy values in general, the lower the value, the better. And so they're automatically ranked for you. This one would be the best one. And then these are the top 20 poses that were generated in decreasing order. To visualize these, of course, in 2D is not that useful. So what we'll do is we'll open up the browser to actually view these structures in the main Mo window. So I'll just move that to the side. I don't need the 2D display, so I'll remove that. And I'll choose a brighter color, maybe cyan, so it pops out here. Okay, so the pink structure again is, and I'll just put the ribbon as well. So the pink structure is my co-crystallized peptide. And then this cyan structure, let's just color the ribbon cyan, that's my first dock pose. So that's the best one, right? The best score, the most negative score. And we can see that it's actually very, very similar to our co-crystallized structure. But of course, I can use the arrows and go view some of the other poses that we got. And so we have this one that's certainly in a different orientation, and so on and so forth. And of course, the scores here drop significantly. Now, in general, we do output multiple poses because there's certainly some uncertainty in terms of which one is the best pose. And so I do recommend not just taking the best pose, but actually looking at multiple of these poses to get an idea of what are the possible binding modes of your peptide or of your protein. Okay, so that's the first way we can dock, which is using the protein-protein docking algorithm. Now, like I mentioned, we can also, and I'll go back to my slides for a second, we can also use protein-ligand docking. The idea here is we're going to use exactly the same framework, the same methodology that we use for small molecules. However, a peptide is certainly much bigger than a typical small molecule that you would want to dock. And so when the docking application generates conformations on the fly for the small molecules, that would not lead to sufficient sampling for a peptide like the one that I have on my screen or the one that I'm going to be using in the next example. So what we highly recommend if you want to use the protein ligand docking application is to pre-generate the conformations of your peptide. And that's possible for small molecules as well. You can always pre-generate conformations and then turn off the actual on-the-fly conformation generation during the docking. So we'll be generating a very good ensemble of different conformations for a peptide and then using those for the placement of the peptide in the binding pocket. So that's exactly what we're going to do right now. So instead of redocking, so in the last example, I just did self-docking, but instead of redocking this P53 peptide, what I'm going to do is I'm going to load in the staple peptide. We'll use the conformational search application to generate a group of a set of different conformations for a peptide, and we'll use that as input to dock to our MDMX receptor using the small molecule ligand application. Docking application, sorry. So, okay, so let me minimize that. So here I'll switch from the protein tag to the general tab, and that's what we're going to use here. But before we do that, we do need to generate the conformations of our stapled peptide. So I'll minimize this and I'll even close this and I'll go ahead and open the structure of my stapled peptide I want to use. So I'll click on the open button and from here I have this stapled peptide that's ready to go. I'll click OK. I said it's ready to go. Actually, I have to prepare it. It doesn't have any charges and hydrogens. So for that, I'll use a quick prep application just like we did on our initial complex. However, I don't want to tether or fix any atoms because I will be generating conformations for this peptide. So I toggled off tether and fix. I'll click OK. It should be quite quick. It's just going to add the missing hydrogens, charges, and do a quick minimization. Okay, so this is my peptide I'm going to be using. As I mentioned, is a staple peptide. So we can see here in stick mode, I have my staple that's connecting both ends of this alpha helix. So what we'll do is we'll actually generate conformations of this peptide. Now, because this has a staple, I know that that central core here needs to remain as a helix. So instead of generating a whole bunch of conformations, some of which will have that alpha helix disappear, right? The alpha helix, if I generate a whole bunch of conformation, will not always be there. But since I know that it needs to always be there, what I'll do is I'll actually generate what's called a rigid body set to actually constrain that section of my backbone to remain alpha helical when I do the conformational search. So anytime you have any input, some prior knowledge about your system, always a good idea to use it to your advantage. So let's do that right now. So what I'll do is I'll select just that alpha helical portion. I'll actually open up my sequence editor, my SEQ here, to select those residues. Let me make that a bit bigger. You can tell that this peptide, which is about 15 residues long, is anchored from this residue to that residue. That's indicated with these blue boxes. You might notice as well that it contains some non-natural amino acids, but that's okay. So I'm going to select just the residues between the two anchor points, like this here. So there's 5 of them selected. And if I go to the main mode window, I can see they've been selected as well right here. Now, I don't actually want to keep the side chains as a rigid body, right? I do want to sample the conformations of those side chains. I just want to keep the backbone rigid. So what I'll do is I'll deselect the side chains. So for that, I'll go to the select and selector options, and I'll use our selection language to deselect. I'll switch this to deselect the side chain. I'll hit enter, and when I do that, you'll notice that now just the backbone atoms are selected for this alpha helical region. The alpha helix continues here, but I chose to just constrain the part between those 2 anchors. Okay, so now that I've selected this, let me just make a set so it's easier to get back to this. So I'll do set create. I'll type helix to create a new set, and as I hit enter, you'll notice there's a second set that's going to appear here. There we go. So now I have a second set called helix, which I can easily go choose again. Okay, so let's go to the conformational search option or menu to launch the calculation. So I'll go compute, conformation, and search to open the conformational search application. Now here, this is the option that I want to use, the rigid body. So this set that I've just created, the helix, that needs to be considered as a rigid body. So I'll select set from the pull down here, and I'll go select my set, which is called helix. And so I won't be generating conformations for that alpha helical structure. That's going to be kept as is, but I will be generating conformations of the side chains, as well as the staple and the ends of my peptide. Okay, so one thing I definitely want to change is this energy window value right here. The default here, as you can tell, is 7, which was chosen for small molecules. What 7 means is that as I'm generating different conformations, I will only be keeping conformations which are within 7 kcal per mole of the lowest energy conformation I've found, right? But for a peptide, I might want to have a bit more conformations with higher energies as well. So I'll increase that to, let's say, 40 in this example. Now, I could also reduce the RMS gradient here. So this is really going to minimize completely each structure. I might want to increase that a bit. And in the interest of time, I might want to decrease slightly the number of iterations that I'm going to run as well. So with that, I'll go ahead and click OK to launch the calculation. And so this is going to open up a database viewer, which will contain all the conformations for my peptide. Okay, so that's going to run. I'm going to have some peptide conformations that are going to appear in the database viewer. Sorry for that. So again, I'm not going to let this run to the end. We'll use some conformations that I've already generated here, but that's just to show you how to set up the calculation. Now, in addition to the mol field, which contains the conformations of my peptide, you'll also notice this delta E field and this E field. I typically don't look so much at the energy field right here, but the delta E, that's telling me what's the relative energy of each of these peptide conformations. So right now, the lowest energy conformation is the first one. That one is the baseline that has a 0 right here. And this is the difference in energy for all of those other conformations. And you'll notice if I collapse this, none of them are more than 40 kcal per mole above the lowest energy conformation, because I set that delta, that energy window to 40. That being said, once in a while, you might see the list actually shrink. So as it finds, potentially better conformations, some of the higher energy conformations will be thrown out. And you'll get a nice ensemble of the conformations. Now, I'm actually going to cancel this calculation. Like I said, in the interest of time, we'll actually use some pre-generated conformations. So just to have a quick look at the output before we move on to the final example, I'll just open the browser so you can see what kind of conformations were generated. And you'll see that in all cases here, there's going to be the alpha helix in the middle because we use that rigid body set. And so it retains that intact. And we can see different conformations of the staple as well. All right, so that being said, let me close this. We'll reopen the MDMX structure now, and we'll go ahead and use the small molecule ligand docking application to dock our peptide to MDMX. Okay, so I'll reopen MDMX. This is the structure I saved a few minutes ago. There we go. This is my peptide. And so I will go get my docking panel back, and we'll go ahead and set this up. So at the top here, I'll use the receptor that I have in my Mo window. That's fine. So that's MDMX. I could use a pharmacophore if I wanted to constrain some of the interactions. So for example, here, as I'm docking my staple peptide, perhaps I always want to keep that interaction. So I could use a pharmacophore to constrain that interaction, but here I won't do that. So where we have ligand here, this is where I'm going to go select my database of stapled peptide conformations. So I'll click on the folder icon. And like I said, I won't use the one that I just generated. I'll use the pre-generated conformational database, this one right here. And as I select that, we can see there's 161 conformations of the stapled peptide that are going to be docked. Now here where it says ligand atoms, I need to change that. It doesn't recognize these as being ligands. So I'll just select all atoms. And when I do that, we can see a 2D depiction of my stapled peptide come up right here. You'll also notice the gear here went green, or maybe you did. That's just because by default, Mo switched to use existing conformations. It detected that this database actually contained conformations of my peptide, different conformations of the same peptide. And so instead of using the rotate bonds application or algorithm, which is the default, or the conformational import, which are two ways to generate conformations on the fly, we won't do that. We'll skip that section. We'll just use the existing conformation. That's exactly what we want to do. That's what I recommend if you want to use this application for docking peptides. Okay, now sometimes I might actually increase the number of poses here. I'll just run it with the default. But of course, these settings, the number of poses that are kept after the initial placement that are refined is set to 30. That's a great number for small molecules. For peptides, again, the search space is much bigger. So I might recommend increasing the number of poses. So here, if I look in the Mo window, what I'm seeing, this gray molecule here, that's one of the conformations of my peptide, my stapled peptide, which was placed and which is now being refined in the receptor environment. So again, I'll cancel this calculation. It would not be very long. It will take a couple minutes only. However, just because we are running out of time, I'll just go ahead and stop this, and I'll show you what the results would look like if I waited a few more minutes. So the results are in this database here. So this is what the output database would look like. So here in this case, we actually ran this with much more sampling than what I did in this setup, right? So I reduced the number of conformations that were generated, and I reduced the number of poses, or I kept the number of poses refined to the defaults. In this example, we actually did much more sampling. And so here we have 50 different dock poses for the stapled peptide on the MDMX receptor. Again, this S field is the final score. That's the GBWSA score that we use by default for small molecules. And so the lowest is the best, and that's the first one right here. And so now I can use the browser again to go browse the different dock poses for my peptide. So I'll go and just change the rendering so it pops out. I'll make that green. And there we go. So this is the first dock pose that we got for the stapled peptide. So you can see if I rotate this way, first of all, you can see that the staple actually fits on this side of the receptor. And if I rotate this way here, we can see that it actually looks very similar to our co-crystallized ligand, right? So it's not the same peptide, but the dock pose, the first one, is very similar, where we have a tryptophan right here, phenylalanine right here. And in this position where we have the leucine 26, it isn't leucine, it's a non-natural amino acid, but it adopts a very similar bound pose, a similar shape to the one that we have in the co-crystal structure with P53. So that's the first pose of the best scoring one, and it looks very similar to our native ligand. So that's pretty good. But then of course I can use the arrows to browse through other poses. The second one again is quite similar. And then as I go through, I'll get some lower scoring poses, which turn out to be quite different than my P53 bound pose. So before we end, I just want to say one last thing, and that's about these columns. So if you look at the output from your docking run, you'll see some columns like these ones here that say PLIF in them. So PLIF stands for Protein-Ligand Interaction Fingerprints. And these are automatically calculated when you do the docking. If you have this option on at the bottom here, generate fingerprints. And so basically these encodes the interactions between your ligand, so in this case, the staple peptide, and the protein environment. Of course, if we look at the numbers, they don't mean much to us, but we do have a tool in the compute menu under PLIF to analyze those fingerprints. So I don't need to generate them. They've already been generated by the docking panel. But if I do analyze, it will take those numbers and actually make sense of them. So here in this panel, each line in the top view here, the barcode view, represent a different ligand or a different bound pose in this case. And on the x-axis here, I have a list of all the residues which make up the receptor or the binding pocket. So all the residues that are actually interacting with one or more of the bound poses. And so, for example, if I looked at let's say the last one here, I could see there's an interaction with the tyrosine here and with the lysine right here. I can also look at the population. So I can see that there's the two residues that are most involved in these bound poses are methionine 58 and valine 89. And finally, I can use this application to pick out the poses that are most interesting to me. So for example, if I click select here, if I only wanted to view the poses that are interacting with methionine 50, that's the one that's interacting with that tryptophan residue, I could go ahead and filter those. And now I can see there's only 6 of them. And if I click on select in database viewer, I now have them selected here. So that being said, I think that's all the time we have for today. So there's a few slides here on the PLIFs, so the protein-ligand interaction fingerprints. I'll skip through these for now because we're out of time, but all of this is in the course material. So feel free to go download that PDF and have a look at the complete course content. So that being said, I'd like to thank you for your participation. As I mentioned at the beginning, we will be sending out the link for the recording of this session to everyone who participated. So that should be sometime towards the end of next week. Meanwhile, if you have any questions that you didn't have a chance to ask, don't hesitate to reach out to us at support@chemcom.com.